Hello, everyone, and welcome to the Bionano Symposium. My name is Ravindra Kolhe. I'm the professor and chair of the Department of Pathology at Medical College of Georgia. And today I'm going to talk about how we are integrating OGM and NGS along with VIA for our research in comprehensive cancer genome. The title is Sequencer Structure, Integration of NGS and VIA for Comprehensive Cancer Genomic Analysis. In the past 5+ years, we have been running multiple research samples from all across the globe, and I think, completed more than 1,000 cases, 1,000 samples, and published a decent number of papers in different phenotypes, including heme malignancies up to solid tumors, prenatal, postnatal cases, and multiple aspects of structural variant research in those samples. Today, I'm going to focus on heme malignancies because I think there's the burden of structural variant has been understudied, mainly related to the techniques we have used in research for heme malignancies in the past. So the other reason is there is an expanding list of abnormalities in heme, and if you just go over the new abnormalities in heme, it's nearly impossible to do that in one single assay like FISH or PCR or karyotyping. So combining a comprehensive bigger panel along with optical genome mapping could be a pretty good way of looking at different structural abnormalities in heme malignancies combined. So at Augusta University, in our research lab, what we do for a myeloid sample, we combine molecular genetics and cytogenetics. The molecular genetics mainly cover targeted NGS panels, about 54 genes, PCR for FLT3, cytogenetics, again, karyotyping and FISH. And as you know, karyotyping has a limitation about the structural variant sizes, and the FISH is, again, very targeted. And then you look at the results and analyze that, the outcome of those assays. And then what we have changed based on what we think would probably be a better way of investigating and researching these heme malignancies is getting a bigger comprehensive NGS panel had helped us to look at these heme malignancies in a lot structural and better way. And then for the cytogenetic aspects of the research, we using optical genome mapping instead of karyotyping and FISH to look at structural variants, including copy number alterations of more than 500 base pairs. And then we compare the results of these two. And we have done some publication in this aspect to look at both and separately at the beginning and combining them together. So this is just a study which goes in detail about comparing these two different aspects and proving the utility of using a broader NGS panel along with optical genome mapping for better research in heme malignancies. So as a proving our concept, what I'm going to do today is going to present some of our research cases to show you how big a difference optical genome mapping it makes in heme malignancies. So this is an MPN case where a karyotype detected a loss of chromosome Y, and the FISH panel was negative for any other markers. Molecular testing identified 3 abnormalities, KRAS, 2 in the KRAS gene, and 1 in TET2 gene. So when we performed the optical genome mapping in this case, in addition to detecting the loss of chromosome Y, it also detected a loss of 4q, which the TET2 gene sits on. But the amazing part of looking at this particular aspect of research is the software. So if you have access to the VIA software, and the VIA software ingests both your optical genome files as well as NGS VCF files, and then we combine them together, and you can beautifully see how well they are how well you can see in a single view both those molecules together, impressively identifying some of the things which have missed in the past. So when you overlay the OGM data with the sequencing in VIA, we detected a compound heterozygous event, basically a biallelic loss of TET2, the deletion of one allele and frameshift variant on the other. So especially with the new way of looking at these kinds of markers, which definitely make changes in the way we do research in heme malignancies. And having them observed in the same view help us make those calls very quickly. This is another case of MDS, where the karyotype detected 2 aberration was a translocation X12 and a 20q deletion. The fish confirmed the 20q deletion, and then we did the NGS panel was positive for MPL. And when we did OGM on this optical genome mapping on this sample, in addition to detecting the structural variant which we found in karyotype and fish, one of the major features we observed was it has a pretty complex karyotype. And then when we overlay both the OGM and the fish data, OGM and the NGS data in the VIA, after we identified a compound heterozygous event in the MER11 gene, which is a participant of the HR complex, and the loss of MRE11 is indicator of an HRD phenotype. So again, not only it adds a pretty significant value to the genomic detection of genomic aberrations by optical genome mapping, but using the VIA software and combining both the NGS as well as OGM analysis in one view makes a pretty significant difference in the way we are investigating and researching these cancer types. There's another case of CLL where we were able to correct one of the structural variants we found by chromosome analysis by karyotyping. So this is a CLL showing several aberrations, but a call on the chromosome 21 was reported at DIC 2121. We did a FISH to confirm by using the RUNX1 probe, and you can clearly see 3 signal, which confirms the 3 copies of RUNX1, hence chromosome 21. And we ran the sample on optical genome mapping. We cannot see the chromosomal gain on the gain on chromosome 21, but however, we saw was a translocation between chromosome 1 and 21. And if we zoom in between that translocation, we can clearly see the translocation and a copy number gain into the structural variant, resulting in breaking the RUNX1 gene, explaining the signal within the fish. Hence, the OGM detected the false positive call with the karyotype in fish. This is another case of correcting the interpretation of structural variant in plasma cell myeloma. In case of plasma cell myeloma, the fish shows a gain of 1 copy of 4p FGFR, 23 out of 50 cells. And when we run OGM on it, the OGM revealed the gain observed in the fish was because of translocation 4.5 with a breakpoint in 4p 16.3. And that particular region overlaps the region of the fish dual fish probe using detecting the FGFR gain. And then the breakpoint of the translocation did not disrupt the FGFR gene, and that hence the fish was called like that. But if you look beyond these things, and this is another case which actually explains why a modality like optical genome mapping is pretty much a game changer in our research for heme malignancies. So this is another case of CLL where the karyotype was not done, but the fish showed a deletion of 13q and deletion of 2 copies 13q. But when we did the optical genome mapping on this case, it showed several additional clones with 1 deleting RB1 in addition to DLEU1 gene. And And then when we look into more detail, the karyotype is more complex in OGM as what was resulted in the fish and in the CLL case. So when we put together all our cases, and in this particular study, research study where we looked at 31 cases and combined a comprehensive NGS panel along with optical genome mapping, and then compared with all the studies which we have done in the past, we have seen a pretty significant additional information which was helping us to do more research on these cases. So not only on one hand, we were able to confirm the copy number variations detected earlier by OGM, but we're able to identify compound heterozygous event per case, cases with of some additional clinical significance for research, and then the risk for changes were also observed. So going back to our original hypothesis of how we should be doing research and how we should be implementing OGM in our heme research, and this is this study clearly helps us to make that call. And then again, I want to add that there are a lot of places which are doing whole genome sequencing, which tends to be an overcall for something like this. And what we like to propose is combining a comprehensive genomic panel with optical genome mapping for your heme malignancy research. So in summary, our research study and approaches shows that optical genome mapping can identify unique genomic abnormalities that include common aneuploidies, sex chromosome aneuploidies, deletion duplications, balance events, and complex structural variations. The other pretty significant aspect of optical genome mapping in research is the significant increase in resolution and accuracy, especially the ability to reveal cryptic and relevant novel variants in myeloid cancers as compared to our previous methods. The other good thing is comparing to whole genome it's a relatively cost-effective approach for using OGM and a comprehensive genomic panel, which is helping us to profile these cancer that not only increasing our yield of oncogenes, leading to improved understanding of cancers. It also is helping us to assess the overall total structural variant burden of this tumor to help us understand the tumor biology, especially how these tumors are separate in different individuals, and then how they can be studied differently. With that, I would like to thank the members in our lab who may help us process these research samples, and our colleagues at Bionano Genomics, Andy and Alka. Thank you.